Welcome students. In this lecture we are going to discuss enzyme purification based on the solubility differences. In last video we have shown enzyme purification based on the molecular size, enzyme purification based on the charge. Okay. Now in this lecture we are going to see enzyme purification based on the solubility differences. Okay, what is meant by solubility? Solubility, it is the process in which protein is separated from any contaminants that may be mixed. Okay, means precipitate is formed in this process and protein is purified on the basis of their solubility nature, or we can say on the basis of their solubility, yes, on the basis of the solubility nature. Okay, now how on the basis of the solubility the enzymes, or we can say proteins, are being purified, that we are going to see in this video. Okay, so there are two methods based on the solubility differences. First is salting in and salting out, and second method is which is the chromatography method, that is affinity chromatography. In this lecture we are going to focus on first process, that is salting in and salting out. Okay, now let's start. It's very simple process. Now carefully listen. What is the process of salting in and salting out? Now salting in means, now suppose first of all it is used in the, this method is used in the determination of precipitation of protein. Okay, this method salting in and salting out, it is used in the determination of precipitation of protein. Okay, now salt concentration, it play very important role in the formation or in the precipitation of the protein. Okay, so it play very important role. Now as salt concentration play important role, it is very important that salt concentration is low or salt concentration is high. Okay, if low salt concentration is there in the solution, see, listen it properly, if low salt concentration is there, it facilitates more solubility. Okay, solubility, we are talking about the solubility. Solubility means what? It helps in the formation of the precipitate of what? Of protein. Okay, so if low salt concentration is there, it facilitates solubility. Means what? It helps the protein to stabilize its structure. Okay, so that is there. And if high salt concentration is there, it cause protein to precipitate down at the bottom. Getting my point? If high salt concentration is there in the solution, it causes protein to precipitate down at the bottom. Once precipitate has been formed, it is going to settle at the pellette side, it's going to settle downside. So how it will settle downside, how protein precipitate will form? When you will increase the salt concentration in the solution. Okay, so ये जो दो methods है, low salt concentration and high salt concentration, this together contribute the process known as salting in and salting out. तो इसमें से salting in क्या है और salting out क्या है? जब low salt concentration है, which facilitates solubility and makes the protein structure in the stabilized form, it is known as salting in. And when high salt concentration is there, which causes protein to precipitate down at the bottom, it is known as the salting out. Okay, so on the basis of the salting in and salting out, our protein is precipitated. That is, when you are talking the precipitation, it is synonym for the purification of the protein. Okay, when you say that we are precipitating the protein, it is related to that we are purifying the protein. Okay, now let's come to the next slide. Now effects of ionic strength and effects of pH, the same is there. Now what is effect of ionic strength and what is the effect of pH? Now first let me tell you about effect of pH. See, protein can either be negatively charged or positively charged based on their condition of the pH. Okay, so the characteristic pH of the solution at which the net charge on the protein is 0 means the condition where the protein is neutral means it has no charge, neither positive nor negative. It is known as the isoelectric point, which we have discussed in our previous lecture. What is isoelectric point? So this isoelectric point of a protein, it is very important property. Why it is important? Because at this point, okay, the protein is very little soluble, very least soluble. Okay, so at isolate, because there is no charge, neither positive nor negative, so it is very less soluble. So different proteins have different isoelectric point. Why? Because some proteins are made up of amino acids and enzymes are protein. Okay, so some amino acids are positively charged, some amino acids are negatively charged. But sometimes condition appear like this where protein is neutral in charge, and at that condition our protein show very less solubility. Okay, so that is very important method. And the method of changing the solubility of the enzyme by using the temperature changes is same as the pH change. Means what? It is somewhat similar to the change in the temperature. That is the effect of the pH. Now the first one was effect of ionic strength. Effect of ionic strength is related to the process known as salting in and salting out. Okay, I have just given you definition of salting in and salting out. Now we will see actual process of salting in and salting out, which is togetherly it is known as the effect of ionic strength. Okay, so let's study this in detail with the help of these three diagrams which has been shown. Okay, so let's start with that. Sorry. So yes, here three tubes are being shown. We'll see about the A tube which consists of three tubes consist of the water, simple normal water, and our target protein is there. The protein structure has been shown in three colors. The first one is black, second one is gray, and third one is colorless protein. Okay, सो आवर टारगेट प्रोटीन मीन्स व्हाट? जो प्रोटीन हमें प्यूरिफाई करना है, जो प्रोटीन हमें प्रिसिपिटेट करना है. हमें तीनों प्रोटीन प्रिसिपिटेट करने हैं, बट आवर द मेन एम टू प्रिसिपिटेट आउट. सी, हमें प्रोटीन का प्यूरिफिकेशन करना है. मीन्स व्हाट? वी आर प्यूरिफाइंग आवर एंजाइम. लेकिन हमें एक साथ सारे प्रोटीन नहीं चाहिए. ओके, हमें एक एक करके सेपरेट प्यूरिफाइड प्रोटीन चाहिए. तो पहले हमारा टारगेट प्रोटीन कौन सा है? आवर टारगेट प्रोटीन इज ऑफ ग्रे कलर. Okay, so we have to first precipitate our gray color protein, and then we will precipitate, or first we will precipitate black color protein, then colorless, and then gray color, depend on the characteristic. But just see the idea how with the help of the process known as salting in and salting out, how proteins are being precipitated, that we are going to see now. Okay, now see, तीन diagrams है. Now what? In that water proteins are there. अभी प्रोसेस स्टार्ट होती है. ओके, हाउ द प्रोसेस हैज बीन स्टार्ट. प्रोटीन विल इंटरैक्ट विद द वाटर. ऑल थ्री फॉर्म्स ऑफ द प्रोटीन विल इंटरैक्ट विथ वाटर. ओके, नाउ हाइड्रोफिलिक अमाइनो एसिड आर प्रेजेंट आउटसाइड, एज प्रोटीन्स आर मेड अप ऑफ अमाइनो एसिड्स. Some amino acids are hydrophilic, some amino acids are hydrophobic. Hydrophilic means water loving, hydrophobic means water hating. Okay, so that is the difference. Now hydrophilic amino acid, these are present outside the protein and they can interact with the water. Okay, and hydrophobic, as they have the phobia of the water, so they are present inside of the protein which cannot interact with the water. Okay, now suppose we add the salt from upside. उस ट्यूब में वाटर है और प्रोटीन है. ओके, प्रोटीन अमाइनो एसिड से बने. दो टाइप्स के प्रोटीन है, हाइड्रोफिलिक, हाइड्रोफोबिक. अभी हाइड्रोफिलिक एज दे आर वाटर लविंग, सो दे आर आउटसाइड, present outside the protein structure, and hydrophobic, they are present inside the protein structure, so they cannot interact with the water. Okay, अब हमने additional क्या किया है? We have added the salt. ओके, सपोज वी हैव एडेड, हम धीरे धीरे सॉल्ट ऐड करेंगे, मीन्स सपोज वी हैव एडेड लो सॉल्ट कॉन्सेंट्रेशन, व्हाट विल हैपन, दे इंटरैक्ट विद द प्रोटीन which will help, they means who, salt will interact with the protein, okay, which will help for the stabilization of the protein. Why they will help in the stabilization of protein? Because we have added the low salt concentration. ओके तो अभी इंटरैक्शन किसका होगा, वाटर और प्रोटीन का नहीं होगा, प्रोटीन और सॉल्ट का होगा, विच विल हेल्प द प्रोटीन टू स्टैबिलाइज इट्स स्ट्रक्चर. And once the protein structure has been stabilized, it increases the solubility. As I told you earlier, low salt concentration, it facilitates the stability and helps the protein to stabilize the structure. Okay, so it increases the solubility, which is known as the salting in. Okay, now suppose in the next condition we have increased the salt concentration. Okay, we have added more salt. So what will happen, salt will start interacting with water instead of interacting with the protein. Okay, salt will start interacting with the water. The interaction between the amino acid or between the protein and the water will start becoming weak. Why? Because salt किसके साथ interact हो रही है अभी, water के साथ हो रही है interact. वो protein के साथ क्यों नहीं हो रही interact? Because they are unable to tolerate the high salt concentration. Definitely they will disturb the stability of the protein. So protein is not going to interact with high salt concentration. Instead salt will start interacting with the water. Okay, so the interaction between the amino acid and water will start becoming weak. As a result, what will happen, water tends to interact with the salt rather than interacting with the amino acid. सिर्फ आपको इतना याद रखना है कि जब आप सॉल्ट का कॉन्सेंट्रेशन बढ़ाते हैं तब सॉल्ट और वाटर का इंटरेक्शन होता है, सॉल्ट और प्रोटीन का इंटरेक्शन होता नहीं है. ओके, दैट इज योर पॉइंट. नाउ प्रोटीन्स क्या हो गए, अलोन हो गए. ओके, नाउ प्रोटीन्स आर अलोन बिकॉज इंटरेक्शन यहाँ किसकी हो रही है, सॉल्ट की और वाटर की. नाउ प्रोटीन्स आर अलोन. नाउ व्हाट दे विल डू, बेचारे प्रोटीन तो अलग हो गए ना, अकेले रह गए, तो व्हाट दे आर गोइंग टू डू, दे डिसाइड दैट यूनिटी इज स्ट्रेंथ, वी विल कम टुगेदर. गेटिंग माई पॉइंट. थोड़ा सा आप उसको हिस्टोरिकल फॉर्म में लीजिए. क्या होगा, अब प्रोटीन्स तो अकेले हो गए, तो प्रोटीन्स डिसाइड करते हैं दैट वी विल कम टुगेदर, unity is strength, so all protein decide to come close and they interact with itself. Instead of interacting, वो ऐसे सोचते कि water और salt का interaction हो रहा है तो हम अकेले क्यों रहे, so they decide to come together and they interact with itself. And water molecule, okay, and water molecule along with the salt start to create shell along protein. अब क्या होता है, प्रोटीन का इंटरैक्शन आपस में हो रहा है. ओके, सो दे फॉर्म लाइक अ बंच लाइक स्ट्रक्चर. ओके, ह्यूज मास तैयार होता है प्रोटीन का और वो ह्यूज मास के सराउंडिंग किसका शेल तैयार होता है, आउटर कवरिंग किसकी तैयार होती है, सॉल्ट और वाटर की. ओके, means they see proteins are interacting, so they provide the outer shell to that huge mass and it will start to precipitate down. as protein साथ में आते हैं, बंच फॉर्म करते हैं, तो दे स्टार्ट टू प्रिसिपिटेट स्लोली डाउनवर्ड. बिकॉज हाउ, बिकॉज सोल्यूबिलिटी गेट्स लेस. यहाँ पर सोल्यूबिलिटी क्या हो जाती है, कम हो जाती है, सॉल्ट कॉन्सेंट्रेशन ऑटोमेटिकली कम हो जाता है क्योंकि प्रोटीन जब साथ में आते हैं, as I told, unity is the strength. So when protein comes together, automatically solubility has been decreased and which helps to stabilize the structure of protein more. Okay, so solubility gets less and proteins are precipitated, that is proteins get separated, they come down. तो नीचे जो pellet है, तो pellet में क्या है आपका प्रिसिपिटेट फॉर्म ऑफ द प्रोटीन है. तो जैसे कि हमने बताया कि लो सॉल्ट कॉन्सेंट्रेशन इट फैसिलिटेट्स द सोल्यूबिलिटी. सो हाउ इट फैसिलिटेट्स द सोल्यूबिलिटी, that was overall process. Okay, so that is been there. If I want to explain you this with the help of the graph, then okay, I'll explain you with the help of the graph also. मतलब जो मैंने आपको theoretically बताया वो मैं आपको ग्राफ के जरिए बताती हूँ, सेम जो थियोरिटिकली पढ़ा है वही ग्राफ के जरिए. नाउ आई विल शो यू. ओके, नाउ okay, let me erase this. Okay, now towards the x-axis there is salt concentration, and towards the y-axis we will indicate solubility of protein. ओके, वाई एक्सिस में क्या है, इंक्रीजिंग सोल्युबिलिटी ऑफ प्रोटीन, और एक्स एक्सिस में क्या है, सॉल्ट कॉन्सेंट्रेशन. ओके, तो आपका ग्राफ कैसे होगा, इट विल बी इन द पीक फॉर्म लाइक दिस. ओके, सो ट्राई टू अंडरस्टैंड. इट इज ये एक ही पीक है, दो पीक नहीं है, सिंगल पीक इज बिन देयर. नाउ सी एट वेरी लो, यहाँ पर क्या है, एट वेरी लो सॉल्ट कॉन्सेंट्रेशन. लेट मी चेंज द कलर. Okay, at very low salt concentration, what is the process? Low salt concentration मतलब क्या है, यहाँ पर salting in है. Okay, there is salting in. Now slowly it reaches towards the high salt concentration. Okay, high salt मतलब जब when we add salt, little salt, that is the salting in. Okay, solubility we can say solubility increase होती है up to the single point, और वो single point ये है. और जब once it reaches the certain point, solubility of the salt drops down, that is like this, which is known as the salting out. Okay, salting in and salting out means what, salts are separated and protein interact with each other, जो हमने देखा, and they form the bunch. ओके, सो दैट जो हमने थियोरेटिकली पढ़ा है वही मैं इस ग्राफ के जरिए बता रही हूँ. एट लो सॉल्ट कॉन्सेंट्रेशन सॉल्टिंग इन, एट हाई कॉन्सेंट्रेशन सॉल्टिंग आउट, and it creates the mass like structure that already we have studied. Okay, so that is all about salting in and salting out. In next video we will discuss our next method, that is affinity chromatography. Thank you.